Okay, so today we are going to hear about biomodels repository. How can we explore biomodels to access reproducible and reusable systems biology models? So my name is Rahman Sharif. I'm a senior project leader at the EMBL EBI based in Cambridge, in Houston, and I will discuss more about biomodels in this talk today with you. Oh, sorry. Mathematical modeling. So I hope many of you are familiar with mathematical modeling. And it's a field of science where you define a biological process with mathematical equations to understand the system. So we have seen that there's an increasing application of mathematics in biology these days. We can use several ways to model a biological system and try to understand the system's behavior. And this modeling is not something new. It's been used since a very long time by physicists to model physical systems. So you can model a physical process and try to understand how that behaves, understand the system. Similarly, you can model a biological process, try to understand the system. And there are several approaches that are used to model a biological system. You can use a simple differential equation, or you can use a logic equation, or you can basically build an agent-based model. And there are many approaches to model biological system. And the models can capture biological system in various scales. You can model, we can model a cell signaling pathway, you can model activation of a protein, or you can model macro molecular processes. You can model an organ level system, or you can model a human level system, or you can model a population. For example, the epidemiological model, which we use to forecast transmission of viruses and so on, which we have seen during the COVID period. They are population level models. So you can model biological phenomena at various scales and try to understand them. And this is commonly used by mathematical modelers across various fields of life science. And they wanted a platform to exchange these models so that they can share these mathematical models they build with each other. And to support that, biomodels repository was established. So in this talk, we will discuss more about the biomodels repository, which is a database of mathematical models. And then I will talk briefly about the reproducibility study that we did to find out how reproducible these models are, because reproducibility is an important part of science. And we want all the scientific finding to be reproducible. And here the question we ask is how reproducible systems biology mathematical models are. Right, to begin with, as I told you earlier, let me just repeat again. Biomodels is a repository of mathematical models of biological system. And it was established about 16, 17 years ago to enable easy sharing, reproducibility, and exchange of mathematical models across various modelers across the globe. So as you can see here in the bottom, there's a link to biomodels. You can go to the link and you land on this biomodels homepage where you can browse various features and quickly access all the mathematical models stored in the biomodels repository. As a mathematical modeler, one can submit their models and share their models with the rest of the modeling community. I can submit my mathematical model in any format, in any approach. It can be an ordinary differential equation-based model. It can be a logic model, petri net-based model, constraint-based models, agent-based models, and so on and so forth. These models can be in any format. They can be in the systems biology markup language, the standard format used to exchange models. It can be a MATLAB script. It can be R script. It can be a Python script. It can be other standards like CellML, FarmML, Combined Archive. So it can be pretty much any format. And all these models in any format can be submitted to biomodels repository and then can be publicly made available through this platform. Biomodels is not a platform that just hosts models for sharing, because you have other resources. We can host your model in GitHub or any other repository, but biomodels offers curation service, which is very valuable. That's what makes biomodels very unique. What is curation? Curation is a process where someone in biomodels team manually verifies the model as reproducible and is doing what it's supposed to do. For example, here, if you have a model of MAP kinase cascade, curator is going to compare the mathematical equation and make sure that they all are consistent. The network is properly represented mathematically, and then the curator is going to encode this mathematical equations in the machine-readable SBML format, then run simulation using tools such as Copasi, and reproduce the simulation results. This is the most important part. So, as a part of curation requires that the figure in the original manuscript where this mathematical model is published should be reproduced. We curate in such a way that we at least reproduce one figure from the original manuscript. This is very important because this kind of gives you trust in the mathematical model, and you know that this model is working and you can reuse it or use it to extend and adapt for your own purpose. So here the key thing is reproducibility, but to enable reproducibility and to make this impactful, we do this through SBML conversion. So what is SBML? SBML is the Systems Biology Markup Language, and it's a format, XML-based format, where you can encode a mathematical model. And there are standard specifications, and there are several tools that support SBML. So I've written 280, I think it's an old number, but there are several tools which allow you to import an SBML model, run simulations, and generate results very quickly. You can import an SBML model into like Copasi, which we use, or Cell Designer, or Virtual Cell, or CopyCell 3D. It can be a MATLAB, and you can import into Python, R, Julia. There are several tools, Cytoscape, which support SBML import. Once you have your model in SBML, you can share in BioModels repository, JetBrains, and there are many repositories which submit, which allows submission of SBML models. It has a very clear structure. It's not supposed to be for human interpretation because it's XML format, but it should be used as Exchange format. So if you build your model in one modeling software, you can export as SBML and then import into other modeling software and run simulations there, or extend the model there. So this allows you, kind of provides you flexibility or interoperability to use the model in various platforms. And this is why we always do our curation or reproducibility of models using SBML models. So BioModels is focusing on SBML, and then we do curation, and the curation aims to reproduce a figure from the manuscript. We have curated many models, and sometimes we focus on certain topics to curate and enrich models in certain areas. So in the past, we have worked a lot on diabetes models, and we have a huge collection of diabetes models, for example, in BioModels database. And this has been published a while ago. And we have also in the past worked on curation of neurodegeneration models. And you can see they're all available in BioModels field, and this manuscript summarizes the collection. Recently, during the COVID period, we started to do curation of COVID-19 models, disease transmission models. The idea is to quickly reproduce the disease of COVID infection transmission models and put them into BioModels database so that any researcher from any part of the world can quickly access it and run simulations and or improve the model, adapt the model for their own local country or city or province, and make predictions there. So this project was actively done, and we have published an archive on this approach. So coming to BioModels, so if you want to get access to any of these models, these are only examples. There are models covering various other diseases, various multiple processes, and so on. You can search for the keyword of your interest in BioModels database and quickly get all the models here. For example, if I'm interested in immune systems, say I'm immune response, so I will go to BioModels database and then search here. As you can see, my cursor, immune response, and this will quickly show all the models which are having these keywords or the synonyms of these keywords, and I can browse them or use the facet on the left-hand side to filter this and find the right model of my interest. If I'm looking for SBML models, I can click on this, or if I'm looking for curated models, click on that, and then this will narrow down my search, and then I will finally be able to pinpoint to the model that I'm interested in. Let's say in the bottom left, you can see there's organism facet. I can choose Homo sapiens model or a mouse model or a general mammalian model. And there's also a facet for disease. So if you are interested in certain disease, you can also search leishmaniasis. You can see in the bottom, maybe too small, but there's a facet that allows you to filter and search models for certain disease. So my models will list all the models which are relevant. You can click on any of these models, go into it, and then try to explore it further. For example, I'm interested in this PD1 TCR signaling model in tumor microenvironment. I can click on that and then I will land on this model overview page. And this page will give a quick overview or summary of what this model is about. It gives me a bit of abstract and gives a little bit of description what the model is about. And on the right-hand side, you see a lot of metadata. So this metadata tells me various information. For example, it says that has taxon Homo sapiens, which means this model is for humans. And then it also tells me this model is about regulation of T-cell mediated immune response to tumor cells and immune response to tumor cells or T-cell receptor signaling. So all these terms you see here because the curator manually annotated these mathematical model with these gene ontology terms. And this whole process occurs in tissue, so you can also annotate the tissue name. And this model is an ordinary differential equation model, so you have this notation for the same modeling approach. And here this model is curated, which means someone has reproduced this model, and you can see all this information here. In the bottom, there are some metrics. For example, it tells you the number of times this model has been reused, or how many times it's been viewed, downloaded, and so on and so forth. All these stats are visible here. You can click on this facet, and then it can show you more information. On the bottom, you can see there's a SBML view. Viewer to the Newt editor. So if you want to get a quick overview of this model, find out what are the proteins that this model contains, you can also click on the icon, and then it will take you to the Newt editor, where you will quickly see all the proteins that are modeled, which include the T-cell signaling, TCR signaling, and the PD1 signaling, and then you can see how they are connected with each other without any mathematical information. Just to get a quick overview. Right. Going next, again, you can see here the same page. So let me go next to show you what else can be done, what else can be extracted. So if I go to the Files tab here, I will be able to download the model. The first file, which is called the model file, is the main mathematical model encoded in the SBML format. I can preview this by clicking on the preview, or I can download this model. Once I download this, I can import this into many SBML supporting software. If they are, and then I can run simulation, I can reproduce the results immediately. Don't have to do any further work, because this is a curated mathematical model. And there are also additional files. Sometimes we also provide a Copasi file. We provide a ZML file, which has information to the simulation experiment that's being done, and this can be then used in conjunction with the SBML file. Right. And if you go to the history tab, then you can see there are how many versions are there, and you see a bit of description on each of these versions or revisions. So biomods allow you to keep track of versions, so you can basically have version controlled submission of your models, and it is possible to download all the public versions from this platform. You can go to any of the version, click download, and you will be able to access this particular version of the model. If you go now move on to the components tab, it gives a quick overview of all the components. For example, you can see here all the proteins and the names, and you can click on them, and it will take you to the UniProt about this protein and give you more and more information. It also gives you a quick overview of the initial concentration of these components used in the mathematical model. Right, the last tab is the curation tab, and this is a very important tab, because this is where we share the figures we reproduced manually. So on the left-hand side, you see all these figures that were manually reproduced by the curator and biomodels. The right-hand side is a figure from the manuscript where you see this original figures. So we managed to reproduce figure 2C, 2E, 2F, 7A, 7C, 7D, and we kind of provide this information in the curator's comment. often it's not easy to reproduce a model. I will talk more about this a bit later, but you may have to change the parameter value or some equation to get this model right, because there could be a typo, there could be error. We fix this during the curation process, and we share this information with the broader scientific community by adding those in the comment section. So, which can be like this equation 3 has to be fixed to such and such thing in order to get this figure reproduced, so on. So this could be very useful, because if you do not know this information, you might be spending or wasting time using the model from the manuscript, which could be wrong. And by using this, you quickly know this is the problem that has been fixed in cases whenever there's an error. But often models are okay, they are reproducible, they do not have any issues, so we can just share the figures that are reproduced in the software that's used to reproduce. But sometimes when they do not, we provide extra information what has to be fixed in order to reproduce this. Just another bit more on search. So if I'm interested in certain modeling approach, for example, the one which I showed you earlier is the ordinary differential equation model. But if I'm interested in logic model, which are based on Boolean equations, I can click on the facet logical on the left-hand side, and the modeling approach, and it's going to give me all the models which are basically logic models or Boolean models. And as I click on these facets, as you can also see in the top in the search, the keywords are added. So if you want to just type this myself, I can do that. Immune response is the keyword which is said, and then I use and then I have this tag called modeling expression : logic model. So there are some hints and examples how to customize your search, and you can read that and then customize the search in order to find the models of your interest. This is again an example. You can click on this model, look at it, and give you a very quick overview. And as you can see here, this is a logic model which you wanted to search and find on certain process. Okay, right. So coming back to here to the summary of this biomodels. So biomodels provides FAIR models, which means findable models, findable through EBI search, OmicsGI, and Google dataset. These models are easily accessible. They are with CC0 license, which means they are free to use. Very permissible. Even academic and industrial persons can use these models without many restriction. The source code is open, and not just the models are free, but even the source code of the biomodels platform itself is open source, which means that anyone can take the source code of biomodels and build another instance of biomodels if you like. And the models are interoperable, which means we here focus on SBML. As I told you, it's a highly interoperable language. It's a community standard, and you can import SBML model into various tools of your interest, and then execute the model and reproduce results. These models are reusable. As I told you, these are curated, validated, and tested, so it gives you a bit of confidence in reusing them. Biomodels is widely used by the modeling community. As you can see, modelers search for data in PubMed and Google Scholar, but after that they come to KEGG. After that they come to biomodels to look for data in a survey. So it's one of the major source of data for mathematical modelers. So you can get a model itself as a data, or you can get certain parameter values or initial concentrations, equations, all those sorts of information from biomodels. Biomodels are also widely used for model dissemination. About 30% of the modelers disseminate their models through biomodels. Of course, many disseminate them through supplementary material, GitHub, personal website, but then the models are not easily searchable. But when you share your model to biomodels repository, the models become easily searchable, findable, and easily accessible. And also there's a possibility for us to do curation, which is going to add more value. So how does it work? So as a user, as a modeler, you can submit your mathematical model to biomodels database. It will remain private, which means no one would have access except you or anyone you give access to. It will be hidden from public. And if you want, you can request access for the reviewer. So you submit your model to biomodels database, you get unique accession number, you put these accession numbers in your manuscript and submit your manuscript to the journal. And when the paper is under review, if you need access to the model, if you need to give access to the reviewers, then you can ask us, and then there's a button that creates a reviewers account. You can share that with the journal, and reviewers can access these models in a private mode, evaluate it. When the manuscript is accepted, we will then publicly release your model. So your model will not be released publicly unless it's publicly, the manuscript is published. And recently we started to also accept a preprint. So if you even have your preprints published, we will be able to release your model publicly in Biomodels database. Right. So, and then whenever the model is submitted, then we have this curation. The model first goes into a non-curated branch and remains non-curated until a curator picks up the model, evaluates the file, extracts the equations, encode them in SVML format, reproduce the figure, and then add semantic annotation like ontologies on the biological processes, gene ontologies, and ontology on mathematical modeling approach, and so on. So all these are done, and the model is then pushed into the curated branch of the Biomodels database. Biomodels data content has been increasing steadily, and we have lots and lots of submissions every year. And these models cover a wide range of organisms, from Homo sapiens to mouse to yeast to E. coli. As you can see, the distribution is varying a lot, and probably there are more submissions on this. Is a bit previous statistics, and we now have more and more organisms. What I show here. And the models cover various biological processes, as you see here. Many of them are corresponding to the response to stimuli. Some of them are biological regulatory process, cellular processes. You can see they cover various gene ontology terms. And these models also cover a wide range of diseases, as you see here. On the top, Alzheimer's, diabetes, Parkinson's are the top diseases that we cover. This is because of the curation studies. However, there are also many other models which are covering other diseases in Biomodels database. If there's any disease you're interested specifically in, just go to Biomodels, search for the keyword, and you should be able to get the information very quickly if there is a model related to that disease available in Biomodels database. Okay, so where do these models come from? All these models are published in peer-reviewed or journals or preprints, and they come from over 300 different journals. And many journals recommend that the authors submit the model to Biomodels database, and you can see them, see a star next to those journals. And most of these models come from these three journals: PLOS Computational Biology, Molecular Systems Biology, and BM Systems Biology in Biomodels database. To give you a quick overview of the content, we have right now over about 2,500 mathematical models, and about 1,050 are curated models, which are mostly kinetic models, which we reproduced. And we have about 1,400 non-curated models, and these are sometimes kinetic models, but majority of them are constraint-based model, logic models, or potential network models. These are the models we didn't have yet time to curate. We are building standards or protocols to reproduce these models, and hopefully in the near future we'll have curated constraint-based models with a new standard called Frog, which we have been recently developing. Biomodels also has a huge collection of auto-generated models. For example, we have Path2 models, which is a big collection of 140,000 models, and they are now archived into 812 bundles based on the different organisms. And we also have a huge collection of patient-specific genome scale metabolic models. And these are 6,750, about 6,750 mathematical models for 21 different cancer types. And each model corresponds to one single patient. All these models are freely available. Anyone can download them and reuse them, adapt them for their own research. Another thing which you want to talk about, which is related to Biomodels, is Biomodels parameters. So modelers often come to Biomodels to not just extract the models, but also to look for mathematical parameters, which are really hard to get from the literature. And we could do that easily because we have models stored in SVML format, which is a machine readable format. So we can easily pass or read these SVML models and extract information such as the species or the entities which are modeled, or the reactions, parameter values, rates, rate equation, rate constants, organism for which the model is, and we can get initial concentration or amount from the SVML model for various entities. All this information can be very easily extracted using the libraries of SVML. this can be then easily put into database, which can be made available through a web interface or an API and allow user to quickly access this information. So we did this and we built this resource called biomodels parameters, which has a nice web interface. You can follow the link to access the biomodels parameters. Here you can search for any protein of your interest, and it will give you all the equations from the models and biomodels in which this protein is modeled, and provide parameters such as if there's a rate constant, rate equations, and initial concentrations of this particular protein of interest. For example, if I'm searching for Cas9, it's going to give me all the equation in which Cas9 is modeled, list them, provide additional information, the rates, and so on and so forth, so that if I'm interested in modeling this protein, if my model has this protein, I can quickly get this information from here. And of course, I should be a bit cautious. I cannot directly use this information. I need to define the context in which this is used in this model and adapt it to my model. It also provides quick access to other resources from parameters. You can quickly go to UniProt to get more information about the protein of your interest, to Reactome to get all the reactions. And you can also go to SubioRK, for example, to get experimentally derived parameters for the protein of your own interest. So this is a very quick overview. There's a huge collection of models that are imported into biomodels parameters, and we have meaningful parameter values extracted from this, quickly accessible to the modeling community. So there are a few use cases I want to show here. So if I'm interested in certain concentration or of co-concentration of certain protein of interest, I can get all the values that are being used to get a range, and then I can scan my model with that range to see how my models behave for a given initial concentration range. As you can see here, I have used an example of a model where we use thrombin concentration as an input to see how the model behaves. Tissue factor concentration to see how it affects the thrombin concentration. And you can also get the Km value for the MAP kinase activation, and see what are the values that are used in the literature, and scan it through your model to see how your model behaves for various concentrations. What you can also do is, from the model biomodels parameters, extract equations and then put them together with another model to build a new model. For example, here I've combined TNF alpha and kappa B signaling model with the ROS and kappa B signaling model. So you have a new model, and then now you can predict effect of components which you couldn't do earlier. For example, you can look at the effect of a ROS or a TNF alpha on SOD. So it allows you to quickly access information from mathematical models. Right, to quickly summarize, these are the two main publications. If you go read through them, you'll be able to get all the information related to biomodels and biomodels parameters. Right, coming to the next part of the talk, which is about the reproducibility study, which I spoke about. Reproducibility is an important part of any science. A recent survey a few years ago by Nature showed that about 70% of the scientists cannot reproduce others' results. I mean, this includes science from biology, physics, chemistry, and so on, and they cannot reproduce results, experimental results from other scientists. And what is also surprising is that 50% of the scientists cannot reproduce results from their own studies. So there is a crisis, kind of reproducibility crisis in the scientific field, which needs to be addressed. And here we wanted to see how reproducible mathematical models are, because these are equations of the end mathematical equation. They should reproduce and they should do what they are supposed to do. Right? We do not expect it to be not reproducible, because equations are equations at the end, and they should, no matter what, reproduce. It's not like experiments, things can go wrong. You can have cells and reagents and instruments. Any changes anywhere can cause deviation in the results and affect the reproducibility, but mathematical equation, we do not expect that. So we want to explore and find out how reproducible systems biology mathematical models are. Before we go into this discussion, I wanted to clarify two terminologies here. There's this concept of replicability and there's concept of reproducibility. Replicability or repeatability is the ability to use the same code provided with the manuscript, with the same software to reproduce the simulation results, right? It's just repeating the same analysis with the same code. However, reproducibility is the ability to recreate the code recreate the model without the original code and reproduce the results in a software different from that's originally used in the initial manuscript. So we were focusing on reproducibility rather than replicability. So we assessed in the study about 455 models from published peer-reviewed articles and see if they are reproducible. What we did, we encoded the model in SVML. As we always do, this was done during the curation in biomodels. We encode the models in SVML, get parameter values from the manuscript or supplementary material, or contacting the author, and run this model to see if the model reproduced. We run in Copasi. If if the original model was built in Copasi, then we use a different software, like it can be a MATLAB, Libsim, SVML, or Mathematica. We use a different tool and try to simulate the result. And we consider a model reproducible if at least one figure is simulation figure in the manuscript is reproduced. We accept minor deviation if they did not affect the scientific conclusion of the study. If a model is not reproducible directly, then we try to fix it, find what the problem is by empirically changing parameter values. That could be a typo. Instead of 0.01, that the manuscript could have listed this parameter 0.1 or 1. So we play around empirically and try to get the model working. If that doesn't work, we contact the authors, and if they respond, then we try to reproduce the model. So the study, to summarize, 455 kinetic models were tested. So we focused on kinetic ordinary differential equation models to be more focused, and these were models published from 1980 to 2020, and they come from 152 different peer-reviewed journals. And this was done as a normal curation activity in biomodels. This is not a randomized sample, but however, it's kind of a major collection of models in biomodels. And there were no random sampling. However, there was this curation priorities in biomodels based on the funding collaborators, which was covering various fields of life science. And the result, this is the important thing. What we found surprising is that about half of the models are reproducible and about half of them are not reproducible, which means we cannot reproduce them directly based on the information provided in the manuscript. And which is surprising because, as I told you earlier, we would expect mathematical models to be reproducible at the end, but they are simply the equations. Unfortunately, it's not the case. When the model is not reproducible, we try to, as I told you earlier, we try to play with the parameter values, try to fix them, fix any mistake in the equation, which can be inferred from the text of the manuscript or from the descriptive diagram and so on, or play around with the initial concentration values, or look for any typos and so on and so forth. We try to fix them and see if we can recover those models. And through this empirical correction, we managed to produce 9% of the models. And then we contacted the authors. When authors respond back, we reproduced 3% of those models. So at the end, we kind of curated more than half of the model, reproduced half of the models, but then about 37% of the models remained non-reproducible, which we cannot reproduce, and we cannot figure out how to fix them. Right. One of the major problems we found is that when we contacted the authors, majority of them did not respond, and about 70% did not respond. Even when they respond, we could reproduce only half of the models, because response means not always means that they provide information. It can be that, unfortunately, the student who did this project is not with us anymore, or I left the academia and then went to industry, or I'm in a different field, I do not have this, I lost my laptop. It can be anything. Right. So all these cases contribute to the other 50%, which we cannot reproduce, 14 models which we cannot reproduce here. And looking at the age of the publication, the proportion of reproducible and non-reproducible models are more or less the same across all the ages. That didn't change. However, response from the authors was quite more from the recent models, so we could reproduce more models that are more recent based on the response from the authors. Right, and we try to look if there's a pattern in the journal. If some journals are good and some journals are bad, unfortunately, there is no pattern, and it's kind of a mixed distribution, and we cannot come to a strong conclusion because for many models, for many journals, we have only a small number of mathematical models, so it wasn't easy to come up with a pattern. So there is no pattern, and we cannot claim some journals good or bad. But we can explore the reason for non-reproducibility, why some of the models are more reproducible, some are not, and we can look into various aspects, and we found that there are 3 reasons why models do not reproduce. There are missing parameter values of the model, or there can be missing initial conditions, or there could be inconsistent in the structure of the mathematical model, or in the equations of the model, or a combination of these factors. However, for majority of the models, we cannot find any reason why these models are not reproducible. It's not clear to us at all. All the equations in the code, as well as in the manuscript, are all correct. All the parameter values are all correct. All the initial conditions are all correct. They're all consistent, but still the models do not reproduce. So we do not have any way to find out what's going wrong. And if the authors do not respond, there's no way, no other way to get these models reproduce their simulation results. So where are these models now, which we worked out in the studies? All the models which we managed to reproduce are now under the manually curated section of the biomodels, but those models which failed to reproduce are in the non-curated section. We haven't flagged them yet. We will soon flag them with a tag that these models are attempted. So we don't want to name and shame. That's the reason why we didn't want to label the tag the non-curated models. However, we don't want the other people to try more, explore again, and then fail and waste more time. So we wanted to put a label, be cautious, or these models were already attempted and we at least failed to use them. And this could also be because we are not perfect and we could have potentially made some error, though it's unlikely because we have enough experience curation. We wanted to be sure that we convey this message in a polite manner. Right, so we have this reproducibility crisis and we have issue. How can we solve this? And to fix this or address this problem, we came up with something called reproducibility scorecard. As on the right-hand side, what you can see is a list of questions asking, are the mathematical equations shared in the manuscript, or the parameters listed as a table, are the simulation conditions provided, are the code shared publicly, are the codes in standard formats such as SVML, is the code is a model deposited in database, it is biomodels or any other repositories, Physium or JDBS online, are the codes well annotated, and are the numerical results shared? These are the 8 different questions. S answer to each of this will give a score of 1. So a model can have a score of 8. So we built a simple reproducibility scorecard. It can be used by either the reviewer or the mod author itself or the editor to quickly score a model. And then what we showed is that as the score increases, the chance of the model to be reproducible also increases. You can see here the total score going from 1 to 8. When the score is 1, almost all the models are now reproducible. When the score is 7 or 8, all the models. So when the score is 1, almost all the models are not reproducible. When the score is 7 or 8, all the models are reproducible, and this is increasing as you see the score increases. So you increase the chance of model to be more reproducible as the score is higher. So we recommend that when you publish your mathematical model, use the scorecard, evaluate it, and see if your model gets a higher score, or do everything that increases the score of your mathematical model. Provide more information, submit them, share them publicly through a repository, and annotate your models better. So we recommend that a model with score should have a score of 4 or more, so that there are a higher chance of them to be reproducible. We tested about 110 models, random models, and we showed that if the models have a score of 4 or more, then with an odds ratio of 4.62, they are highly likely to be reproducible. So this study has been, it's also published a couple of years ago and nicely summarized. And when we tweeted about this work, and it's one of the important topic discussed, shared in the community, as you can see here, we got 100 likes. of our tweet is sort of the most liked and shared tweet of biomodels, and it created definitely an impact in the field. Okay, coming back to biomodels. So biomodels is interested in promoting reproducibility. We share reproducible models, and we make sure the models are curated properly, which includes reproducibility annotation, and shared with the wider scientific community. So this is important so that the modelers can reuse the models in biomodels. As an example, I can quickly show you, and a modeler can pick a model that's already in biomodels and reuse it. You can see here, some authors do that. So a Proctor 2005 model was then reused in or repurposed in Proctor 2007, Proctor 2011. However, this model was then also further used by Tang to 2010, and so on and so forth. So you can see, like any other scientific discovery or scientific manuscript, is such you generate a knowledge which is then used by another manuscript or another scientist to develop further. So similarly, a model built by one researcher can be again used by the same researcher, or when shared in public repositories by models, can be reused by another modeler to extend and repurpose to repurpose the model for their own scientific purpose. So the model can keep evolving to address different biological questions in this and provide a broader impact when the models are shared publicly through biomodels database. So as a modeler, you can submit your model and increase this impact, but also reuse the model that's already in biomodels database. So as a modeler, what you can do is quickly search and retrieve appropriate models from biomodels database. You can evaluate if the model is suitable for your investigation or research. If it is, then you can either use it as search, or you can extend or modify it to accommodate the input-output, and then you can evaluate your mathematical model experimental findings, the iterative process. So you have to improve the model again if there is any discrepancy. Finally, you can apply this model to solve your problem or guide you to do new experiments. Coming to almost the last slide. So basically, by providing the models easily accessible, making them findable, accessible, and reusable, you're basically standing on the shoulders of the giant, which means you can reuse a huge collection of existing models instead of having to rebuild them again from the scratch. This will accelerate science. So by doing this, supports model discovery and reuse. The modelers can benefit from easy discovery of the models, and the models are in standard format so that they can reuse them in various tools of their interest. The curation enables reproducibility, so that the models can reuse their models with confidence, and the semantic enrichment is very valuable because it improves the findability, but also helps the model to unambiguously identify various components of the model. Right, with this, I would like to end my presentation. Thank everyone for listening, and everyone here for their contribution to the biomodels database and the funding bodies. And I'm happy to answer any questions. Meanwhile, you guys can explore biomodels using clicking on the link below. And if you have any questions, feel free to ask, and I will answer them. Okay, so if there are not enough questions, then I can go to the demo and then show you how to browse models to biomodels.
Yeah.
Okay, here is the biomodels web page. You can click or type the biomodels URL. And then maybe you can also share that via chat if someone wants to click that and explore. Okay, you did already. Sorry.
Yeah, I did. I can do it.
Okay, and here you can search for a keyword of your interest and find a model. So does anyone have any suggestion? Is there anything that you guys want to search? If not, let's say I will search for, let's say, cancer. If I search for cancer, I should get all the models related to cancer. You can see here about 332 different models. Just make it a little bigger. Oh, right. And I can explore this and then find the model of my interest. On the left-hand side, you can see various facets, as I showed you. If I'm interested in curated models, then I can click on this and it will show only those which are curated. Curated models are those which are reproduced. As I told you earlier, at least one of the figure from the manuscript has been reproduced. And we can use these models quickly, which means I can import this model into the database of my interest. I can further filter down. If you want to use ordinary differential equation model, I can click on this, or if I need a delay differential equation model, I can click on this. If I need Homo sapiens, I will click on this, just filter Homo sapiens models, for example, here. And I can then characterize them if it's lymphoma, leukemia, breast cancer, non-small cell cancer. There are also other annotations which provides you further narrow down search. And there are also gene ontology terms like cell death and so on and so forth, which you can use to further narrow down the model of your interest. So for now, I'm going to just choose one of these models, let's say cancer and immune biomarker. I'm going to open this in a new tab and go here and show you a bit more on this model. So as you can see, there's a quick overview, and you can see on the right-hand side the metadata. This is the immune response to tumor. This is in ovary. This is the ovarian cancer probably. As you can see here, is ovarian carcinoma model is a differential equation model, and they get all the information on this model abstract and further information here. If I go to Files tab, then I can quickly download the SBML version. If I click Preview, as you see here, I don't know if you could see, you will get a preview of this SBML file. This is not supposed to be human readable, so it's totally fine. This is an Exchange format. You can import this into any of the modeling tool very quickly, and then you should reproduce the simulation result. Right, so I can click on then history. You can see there are 7 versions, but only the 7th version is publicly available. If you go to the components tab, then you can see what are the components. Then you have mucin, lymphocyte, malignant cell, interleukin 7. These are the main components of this mathematical model. And if you go to the curation tab here, you can see a figure, which is figure 3a, which that's being reproduced from the literature using the Copasi tool. So this is a very, very quick overview, but if you want to get more on the network connection of this model, you click on the SVGN view. So this will open the model in the SVGN format. SVGN is Systems Biology Graphical Notation format. As you can see here, you see how the different components of this model are connected. So pretty much that's it about quickly accessing the biomodels database. You can browse more at your own time. And if you have any questions, feel free to write to us, and we'll be happy to help you. If you are a mathematical modeler, and if you have your model published, then submit your mathematical model to biomodels database. We might be able to do curation. And this is quite good because it increases the visibility, reusability of your model. And there are reports that shows that models from biomodels are more cited. So if a model is curated, then the chance of them being cited increases. So to increase the visibility, citation of your model, do submit your model to biomodels database.
We have one question here. If we can learn basic mathematical modeling course here. So I guess if you could suggest some courses or something, if somebody wants to learn mathematical modeling.
Yeah, I think, of course. So probably you can go to the DBI training resource, and then you probably can access the content of the Mathematics of Life course. We run a course every year on called Mathematics of Life. It gives you a bit of overview of various modeling concepts. You should be able to learn from there. And of course, we run also this course either virtually or in person. Hopefully this will happen next year. This year we didn't run it, so you can participate in this course which we run. If it's in person, it's a residential course in Cambridge, and that's a good opportunity to learn mathematical modeling. And that's primarily aimed for those who are highly interested, but also experimental person not having enough background. So it's mostly for beginners. So it would be a good course for those who wanted to get an overview of mathematical modeling and various approaches.
one question which came in. Is it possible to search for models using the name of a protein to find which model this protein fits in?
Yes, I think this is a very good point. You can search for a protein. Let's say I'm searching for cyclin. If I can search cyclin, and then of course you see all the cell cycle models you can see here. So you can search for a name of with the name of protein, and then you should be able to find all the model that has that protein. And you can go to the UniProt here, the facet, and then filter out and click on those protein of your interest, like so APC, second dependent kinase 4, V1, E2, for example, FBOX only produce. So there's a facet here where you can filter your search to find protein of interest. And if you want to focus on a certain small molecules like calcium or ATP, you can also click them here, and then you should be able to filter out your model to find the right model of your interest. That's definitely possible.
Thank you.
Yep. Just a quick remark, modeling is quite broadly used by researchers who are into fundamental science, and also is quite commonly used in the pharmaceutical industry. So when you have a drug and you want to know what's the concentration of the drug that needs to be prescribed and how often the drug needs to be prescribed, you need to build a mathematical model based on the pharmacokinetics and pharmacodynamics of the drug. So basically looking at how the drug is absorbed by your body and how the drug is metabolized by the body. By doing this calculation estimation, you need to mathematically estimate how much drug you need to insert and inject, and how often you need to inject to maintain certain dose or concentration in the body for the drug to act or cure a disease. So it's quite commonly used in the pharmaceutical industry as well. And to learn mathematical model, one good way is to directly get the basics and then directly download the models which are freely available, and then import them into tools such as Copasi and play around with the model. So you can explore the model more, and then learn about modeling. That could be approach to about that. And someone said, who's a microbiologist, of course, there are lots of models of microorganism, and these days genome scale metabolic models of microorganisms are widely used, getting very common, and they are used for also metabolic engineering. So you can basically get those models from biomodels, download them freely, import them into tool of your interest, explore them further, and use it for your research.
So we have one new question. What are the most used programming languages to simulate mathematical model?
I don't know what's mostly used. I think there are many which are quite common. The one which we use is Copasi. If you are a beginner and if you want to, you have a nice user interface, you can download Copasi, and there are tutorials how to use Copasi. And just download the SVML model, import them into Copasi, run simulation to generate the figure, and then you can play around with the model very quickly. But if you want to help, if you have a coding background and you want to do a bit more sophisticated modeling or sophisticated analysis, or maybe very quickly some analysis, then you can use Tellurium, which is a Python package for mathematical models. Download SVML main file from biomodels, import them into Tellurium, and you can quickly play on the model. And Tellurium also kind of is linked with Antimony. So Antimony is a very simple way to encode model. So you can write equation or write a code for a model in like 2 minutes and run simulation with that. But those are all command line based as well. It depends. And then you have Cell Designer, which has a very nice interface to build models. When you have biological pathway, you can represent the biological pathway in Cell Designer and then convert that into mathematical model and simulation. You can use MATLAB as well. MATLAB has SimBiology toolbox, and it also has a nice interface. You can use that. R also has a package. So there are many tools that supports mathematical. And there's this agent-based model, which is a different category of models. What I was talking about mostly was order differential equation model. Then you have logic model. They have different set of tools. It depends on interest. You can pretty much use various resources and run SVML model. Once you export model from in SVML format, you can import any other tool that supports them and then run simulation there as well. So that's another question. And do you have a model example of models used for target discovery? Yes, I'm not sure, but that could be. I haven't explored. That could be some models, but we have many PD models as well. For example, just used for the drug dose optimization stuff. So that could be models for target discovery. So any model, if you can investigate, use to investigate effect of a target, potentially it can give you some hint on target discovery. There are big signaling models. You can do in silico experiments where you can perturb various components, which could be potential targets, and see how they affect the disease progression and so on. So that could be some models. I'm sure that kind of are looking for using these modeling approaches to discover targets for specific diseases. There are a few more. So you can maybe look for drug target as a keyword and then see how many models turn up, and that could be one way to explore quickly and see what's there.